Hello everybody, I'm Salvatore Cosentino from Iwasaki Lab at the University of Tokyo, and today I will give a talk titled SonicParanoid2, Machine Learning Driven Integration of Bidirectional Best Hit and Protein Domain Analysis for Faster and More Accurate Orthology Inference. The talk will be basically separated in two main parts. In the first part, we try to address the problem of scalability due to Oversold alignments in graph-based orthology inference. And in the other one, basically, we would try to infer orthologs without using bidirectional best hit and find ortholog pairs that bidirectional best hit alone cannot find. So, talking about scalability in graph-based methods, basically, if we have two proteomes, A and B, with N and M proteins, if we want to find the orthologs, the first thing we need are the Oversold alignments. These can be separated in two main classes, the interproteome alignments, which we use to find the main orthologs. And these are alignments between two different species, like, for example, AB in here or BA in here. The intraproteome are alignments of a given proteome against itself. These are the most consuming, and we use it usually to find imparalogs. Graph-based methods, among these there are, of course, SonicParanoid, but also Imparanoid, Proteome, and many others. The scalability, if we look just at this table, it's very easy to see how it is influenced by the interproteome alignments. For example, on the left we have the number of input proteomes, and while the intraproteomes grows linearly, the interproteomes grows exponentially. So after a handful of proteomes, even let's say 100, 99% is interproteome alignments. So basically, the idea is that if we can reduce the execution time of these parts of this kind of alignments, then we can save some time for orthology inference. And alignment tool, local alignment tool, basically they usually advertise a big O NM or big O N square time complexity. But in general, there are many tools. There is, of course, BLAST, there is MM6, Diamond, just to say three of the most famous. And usually the most expensive step, for example, in this case, as we see in the second point, is the first search in the database, in which we find potential, the tool search for potential hits. For example, MM6 is called also prefiltering, for example, if I'm wrong. But the important thing is that this time complexity basically assumes that the number of queries is much smaller than the number of sequences in the database. But in reality, of course, there are many things affecting how long it takes, right? What I want you to keep in mind is that, as we said before, if this is not satisfied and much smaller than M, basically, if this is true and we invert the alignments, for example, BA instead of AB, then the execution time could be very different. This also implies that when the two proteomes, let's say again A and B, let's say are very close, let's say similar sides or very closely related phylogenetically, then even if you invert BA or AB, the two alignments should be almost the same execution time. As we said, we use graph-based, usually use bidirectional best hit. In the bidirectional best hit, basically, if we have A and B from two different proteomes, in order to be orthologs, basically there are two conditions that needs to be satisfied. The first one is that B is the best hit for query A in the alignment AB, and A is the best hit for query B in the alignments BA. Now we just need to remember that, I want you to remember that when we basically process the alignment, let's say AB, for example, the first thing we do, we apply a threshold to the hits, right? And so if we remember this, basically it happened, we know that some query, for example, it could be that in the alignment AB didn't actually appear because it's matching nothing. So if we remember this, when we do the alignment BA, if you think about it, it's maybe a waste of time to actually search for A in BA if we know that it cannot be an ortholog based on the bidirectional best hit. So we define this very simple concept, which is the essential bidirectional best hit. And basically what we do, we first perform, after assuming we know already the first alignment, let's say AB, in which there are pairs, again, of hits AB with a given bit score, we only keep, basically, we create subsets only keeping, for example, A from queries A from A, such that there is at least a target B with a score above our threshold, right? And same we do with the target sequences, in which we only keep sequences so that there are hits for a query A such that the score it's higher than the threshold. Remembering this, then if we redefine the BBH, which we call essential BBH, basically the first clause would be the same. B is the best hit for query A in the alignment AB, but A is the best hit for query B in the alignment essential B and essential A, where essential B and essential A are subsets of the original input proteomes. What does it have to do with the alignment execution time? So this, I'll give you now a practical example. So basically, in this example, we have Plasmodium falciparum, which is the malaria-causing parasite, and Chlamydia trachomatis, which is a bacteria that causes trachoma. They are very different in size, in proteome size, in protein size, and also phylogenetically. Now, if we align first Chlamydia trachomatis with the falciparum, the execution time, in this case I did it with MM6, the most sensitive, it was about 43 seconds. If we do the other way around, the alignment takes much longer and is about 6 times. It takes 6 times the time of the original. Assuming we know already the first alignment, right, the fastest one, and then we create the subsets of sequences as I explained in the previous slide. Then the total execution time, because we know already how long it takes the first one, the total execution time, it actually takes is 5 times faster. And the input we use in the second alignment compared to the original one, in average, is less than 50%. So basically, how do we do this prediction then? So the first thing we do, we predict which is the fastest between the alignments between AB and BA. The other thing we do, we simply align AB with the whole input, and then we create our essential set, essential A and essential B, and we align essential B and essential A instead of the complete set BA, and hopefully save some time. How do we predict the fastest alignment? We train the adaptive boosting model, and we performed 16,250 alignments, which are then the training samples. And these training samples, the features are very simple information that we gain regardless from when we process input in SonicParanoid, which are in this case the sequence counts for both proteomes, the sequence counts difference, proteome sizes, and mean protein length. Then for training the model, we perform tenfold cross-validation. We obtain 97.15% accuracy, average accuracy cross-validation. We also tried to build the model and use more, let's say, more different kind of features like molecular weights, the weights of the amino acid basically, but didn't gain much in accuracy, and also this require extra computation, which we want to avoid because in the end we want to save time, right? The way we label the sample is very simple, of course. When we run the alignment, in this case 16,250, if AB is the fastest, then we put it as fastest, otherwise we label it as lowest. The problem is that in some of this alignment, basically, if A and B are very close, very similar also phylogenetically, basically it can happen that if the system, because we do it in parallel, if the system is very busy, it could happen that sometimes AB is faster than BA, but in another run, it could be that BA is the fastest, right? So what I'm trying to say is that in some cases, there could be some noise in our training data. The only way to reduce this is to run, in my case, I'm using MM6 at the most sensitive settings, which would mitigate somehow this effect I'm talking about. So the training set and the data set. So for the training set for the AdaBoost model, we selected 250 reference proteomes from UniProt, 200 of which are eukaryotes. So it's kind of big, there are many eukaryotes. And for the test set, we are using the benchmark, the 2020 version of the Quest for Orthology benchmarks with 78 proteomes, 48 of which are eukaryotes. We selected the QFO also because it contains organisms that are both very distant, but some are also very close, and also because we can use it for benchmarking the quality of the prediction that we do in the end using the benchmark service. And I actually take the chance now to thank all the people from the Quest for Orthologs for creating this very useful tool to the community and to me, because they saved me lots of time. So thank you a lot. The hardware we used, unless I don't say anything different, is we used basically 128 CPUs and 1 terabyte of RAM. So it's a kind of a big machine, strong machine. And when I talk of sensitivity about SonicParanoid, I usually mean the setting of SonicParanoid, which there are 4, from fast to most sensitive. But I will also show results for Diamond, and these of course have different naming for the setting. So with the MM6, so if any of you listening maybe wants to know which corresponds to which, we can use this table later. So how much time do we save in the end? So on the top in here we have the results from MM6-2, and as we can see in this box, basically the more we increase the sensitivity from fast to most sensitive, the more we save time, and we save up to 44% of the time that would take using the complete alignments basically. And we can see this column in here. Basically this column in here shows the average size that we use in the subset. So basically 33% means that if for a pair A and B we create the subsets A and B, let's say for A it's 20% of the input, and from B we use 30% of the input, that will be 25%. The average is the average among all the pairs. So basically the lower it is, the more we avoid alignments. That's the meaning. And then we go to Diamond. So Diamond also, okay, Diamond, you need to know that it's much faster than MM6 and it behaves differently. As we can see, at least up to the sensitive mode, it does increase a little bit the time we save, but then it falls on the most sensitive mode. I'm really not sure what is going on. We are still saving some time, but I will take a look more closely to what is maybe happening in there. And as we see the average of the reduction that we have, it's not so different from MM6, so it's kind of safe. And where do we save this time, basically? Where do we do this reduction and then save time? So basically in this plot, on the X, we have the mean average amino acid identity, AAI, which tells us basically an idea of how close each pair of proteome is. Each point is a pair of proteomes. And on the Y, we have the mean, as I said before, for each pair, the average reduction that we have on the original input. So on the left, what is labeled as mixed, basically are prokaryotes aligned to eukaryotes or vice versa. So as you would expect in here, as they are very distant phylogenetically, basically we avoid a lot of sequences. The subsets are very small. We can see that in the prokaryotes in this block, there are all more different distribution. And what happens with MAGs, I think actually that for this, what we are doing now, it will be very important for MAGs, because MAGs, you can be very different, low quality, good quality mix, so could save a lot of time, I think. And when we go to the eukaryotes, this is nice. This was a good idea to use the QFO. As we can see in here, for example, the reduction is very low because these are very likely, I didn't check by hand, but I would guess they could be, for example, Homo sapiens with gorilla or something like that, but some organisms that are very closely related. So the reduction is very low. But the things that you need to remember is that it's not slower than the original alignment anyway. So that's very important. And another thing that I checked is if I really need to predict the fastest pair, how much does this affect the total time we save in the end? So basically what I did, I took the model, I did a run in the most sensitive mode, and I inverted my prediction. Basically, if my AdaBoost model, which was 100.63 accurate, I basically invert the prediction that it gives. So the predictions are only 84% are correct, so they're mainly wrong. So the result is that the alignments were 3 times slower than when using the correct model. So we need to use the correct prediction model in AdaBoost. And now is the most important thing. how does this affect the final quality of the predictions that we have? Again, we use the benchmark from the QFO, and I'm showing on the top, this is on the left, the eukaryote and the Luca. In order to read the results, I only want you to remember that the markers, the circles are MM6, and the diamonds are, well, diamond, and the empty markers are basically those with the reduction. So if you look at pairs, for example, if you look at this, you see that the difference, these are both MM6, for example, in the fault, they are very close, and all of them, like here, they are very close. They tend to be all very close. And when you go to very small data sets, for example, in here, in gene ontology test, which the dataset is very small, they tend to overlap, which makes a lot of sense. But the message in here is that the accuracy is not being affected negatively, and that's the most important thing, actually. So what did we do in the first part, basically? So we reduced execution time for all Oversold alignments, simply avoiding unnecessary alignments. It seems there is no negative effect on accuracy, and it could be used by other graph-based methods, which I think is a nice thing. And it works regardless of the alignment tool, albeit sometimes in a little different way. What remains to be done? I also want to do this with BLAST, because BLAST is the slowest among the three that we have mentioned so far. And I think it would be nice to do it for BLAST, see how it works in BLAST. And then, as I said, with MAGs. Actually, I did a run with 2000 MAGs, and it took 2.4 days. There are 4 million alignments, so it's really a lot. And the average reduction I obtained is about 30%, which is really, it's quite a lot, right? So, but I really didn't have time to do the normal alignment, so I'm not sure. I will confirm. I will have to do this run. Sorry, I didn't have time for that. So can we use it? Yes, actually, I implemented this the first time 2 years ago, and actually the running version of SonicParanoid, it's already including this part, but by the end of July, I would like to, I'm planning to update SonicParanoid with the newer model and also support Diamond for people who want to use it. Now we move to the second part. So in the second part, basically everything starts with BBH, right? So bidirectional BST is very nice, but it has its limitations. So basically, one of the limitations, for example, is when there are very distant orthologs, or in general, when there are organisms, when you are comparing organisms with lots of duplication, for example, plants, it can be possible that some of these orthologs are missed. So we need a different source of information. Domain architecture is basically a possibility, and there are studies that suggest that domain architecture, when domain architecture is basically conserved, let's say they're very similar, it can be that the function is also conserved, so it could be used as a proxy for orthology. There are not many tools yet that predict based on by analyzing architectures. One of these is Domainoid, which was created by the same authors of Imparanoid, and actually I'm borrowing one figure from their paper to show you what can happen with full-length approaches and how they can be missed, how the orthologs can be missed. For example, this is an Homo sapiens protein with 2 functional domains. These unknown are just interregions with no PFAM hits. They are using PFAM. And so in Escherichia coli, this protein is split in 2 functional domains. So the BBH we use, for example, SonicParanoid probably would just match one of the 2 as bidirectional best hit, but it will very likely lose the other one, right? So we need to do a different way in order to find these orthologs that we can't. So in here, basically what we would do now, what we try to do is to think of architectures as phrases as with words, right? And use a neural network to try to compare these phrases and predict orthologs from these phrases. Since I cannot give you a very detailed explanation, I will try to give you an overview as much as possible. So basically, the method is separated in 2 main phases. In the first phase, basically, we predict the functional domains, and we train the model, right? In the first step, for all the N proteomes, we search for functional domains in PFAM, and this figure basically shows the percentage of hits. So it's the coverage, if you want, of the proteome. For how many queries we have at least one hits. That was to see how much we have. This has to be limited because of the limitation of the database, right? It's average, I think, 50%. Then we extract the architectures. For example, this is an example, very simple, too simple architecture. Actually, it's just an example. And these two are interregions for which there is no PFAM hit, and they can be of different lengths, right? And this is very important because we will actually be using it in our model. I also want you to keep in mind this on the red boxes, which are the architecture sites, which is the number of parts composing the architecture, the query length, and the query coverage. The query coverage basically says the ratio of the length of the protein, how much it is covered. So in this case, it's about 40%, right? Then when we look at these architectures, basically, so on the X, we have the length of the proteins. And so when we increase the length of the proteins, we have that, as you would expect, the coverage goes down, the query coverage goes down. And when we look at the architecture sites, when it gets longer and longer, we have the architecture gets more and more complicated with more different domains and very high number of, let's say, words, right, which are actually functional domains. Again, forget about functional domain, think about words. Now, if we look at this architecture, we create documents, basically, in which we have words, right? And we see also this number. These are also words. Basically, we are encoding this missing interregions, this uncovered interregion of different lengths as different words, right? So that would be part of the vocabulary. Then once we have this vocabulary, we train our doc2vec single layer neural network, where doc2vec basically is an extension on word2vec from Google. And it's basically what it does in a very simplified way. When you have the trained model, you give a phrase, in our case is an architecture, and it will give us a vector, a numerical vector. So if you remember what I said a few seconds ago, there are these values that we use, and this we use it for prefiltering. Why do we prefilter? Basically, if we have two proteomes, right, and we have a given number, N and M architectures, not all of this can be, of course, ortholog. So we don't want to analyze all of them in detail. So we prefilter them. And one way to prefilter them is this free information I said before. For example, if I have two architectures and one has only two, the architecture size is 2, only 2 functional domains, and one is 15, maybe it's not worth it to compare. So that's how we prefilter, basically. Then once we have these prefiltered architectures, what we do for each of these architecture, we take each pair and we compute the cosine similarity. The cosine similarity, basically, it takes in input 2 vectors, right? And it will give us a similarity between these phrases. When it's 1, they are the same. When it's lower, they tend to be different. In our case, we only keep cosine similarities higher than 0.5 for now. Then we create a matrix. For each pair of proteins, we create a matrix in which we have cosine similarity in each cell for each pair of architectures. I will explain this a little bit later. And then we predict the orthologs. And once we have the domain-based orthologs, we merge these domain-based orthologs in here. We merge them with the alignment-based orthologs. So how do we predict this? How do we select this domain-based orthologs? So as I said, we fill a matrix for 2 proteomes, right? And for each architecture, even though there are 1, 2, 3, for each architecture, we compute the cosine similarity. And then first we analyze this matrix row-wise and select the max. So we select the max cosine similarity. And then we do the same thing by column-wise. We select the max for each column, as you can see here. And then we overlap these 2 matrices. And then when we have the overlap, basically we kept those as orthologs. Then if we just take a look of what we predict, the domain-based, we predicted 7.24 pairs of orthologs, which is half than SonicParanoid in the most sensitive mode, which takes, of course, much, much longer. And 8.3% of these are actually contained in the most sensitive graph-based SonicParanoid. But the message in here is that we are predicting pairs that are not predicted by the by the graph-based algorithm. Then the most important thing, when we merge this, we look at the results. We are showing Luca because Luca is basically the last common universal common ancestor, and is the biggest data set. It will use most of the pairs that you give in input to the method. And as we can see in here, this is the graph-based, in the most sensitive mode, the black one. You can see that in the green one we have an increase in the number of completed trees, but we also have a slight increase in the RF distance. So basically we are introducing some errors. This can be solved basically because right now we are basically doing the union in a wave, but we are developing an algorithm that will actually check if this ortholog was already found, for example, in the graph-based, and depending also on the cosine similarities, and do a better clustering and skip some orthologs, split clusters, and so on. Execution time and scalability. So this that I'm seeing now, of course, the ortholog, the architecture-based analysis took about 40 minutes. It is more scalable than the Oversold Alignments. The reason is because the profile search for n proteomes, we do it only n profile searches. And also for training the model, basically, because the architectures, the domain architectures tends to be repeated, once you keep adding proteomes, basically you will have, your vocabulary will reach a plateau, so will, of course, the training time. Another important thing is that the training is done, the training of the models is unsupervised, so it's done on the input, no pre-trained model, okay? And then we want to keep it usable if possible, so user should be able to choose if you only use the graph-based or the domain-based or the merge or the hybrid part, as we will call it. So in conclusion, in this two part, we did two things. We reduced execution time in Oversold alignment up to 45% by simply avoiding unnecessary alignments. The nice thing is that it doesn't seem to affect negatively the predictions, and then I think it could be used by other graph-based methods. For the domain-based, we have increased the amount of orthologs we are predicting when we merge with the graph-based, and I think the scalability is nice, and I actually can improve it even more. Now it's time to thank you. I hope I was not too fast or too slow, and I want to thank you despite, because for some of you maybe it's very late or too early in the morning, so I hope you enjoyed the talk. Then I want to thank the organizer of ISMB and ECCB, and especially EvolCompGen organizer for giving me the chance to talk in this nice venue. And then of course I want to thank Wataru and all the people at Iwasaki Lab for letting me do my research. Has been very nice the last couple of months and enjoyable. So thanks a lot, and I'm really looking forward to all your nice comments, which I'm sure that will boost my research. Thanks a lot, and have a nice day or evening wherever you are.